We have an extraordinary keynote speaker in Jonathan Rothberg, who is a serial entrepreneur and inventor, was introduced to the department and myself by Joel Bader, who he worked with some time ago when they did 454 sequencing technologies, which was purchased. And then Jonathan went on to do Ion Torrent, which is a fantastic sort of, is it called third generation sequencing technology or something? What Jonathan is up to now is he has a fantastic incubator called 4Combinator up in Connecticut, in which companies are kind of sequenced, I would say, or in terms of their maturity. And so wild ideas come in the front end, and they get developed into companies that come out the back end. And I think the plan is to have 4 at once. I don't know if you'll talk about 4Combinator. And he, as always, is after high quality input into the technologies he's developing. And so he was very unabashed at saying that if he was going to participate in the program, he wanted to take away some of our best talent, right, in terms of the students to come up to 4Combinator. And that is a great outcome, and I hope that's achieved. And thanks very much for coming down to talk to us today, Jonathan Rothberg.
Thank you. And I was quite inspired by the presentations. And those of you who made presentations and love your work, send me an email. Okay, let's get going with my talk. I'm going to start a few summers ago. In Germany, many of you may have noted a few summers ago, over 900 people were rushed to the hospital. Over 50 of them died. And the cause of that was a strain of E. coli that the world had never seen before. And one of the interesting outcomes was that the microorganism that was making these people sick and ultimately killing a number of them was decoded by DNA sequencing. And what I'd like to do today is tell you, how did we get to this point that we could understand an outbreak by decoding a genome of a microorganism on a $99 disposable chip. That genome was decoded over about 2 hours. It was put on the internet. A postdoc in Australia decoded and analyzed the genome, noted it was resistant to 14 classes of antibiotics. Other groups used the sequence in near real time to make polymerase chain reaction probes and create a diagnostic that would allow people to screen food sources for this microorganism and, through analysis of the genome, understand what antibiotics to give the patients and which antibiotics not to give the patients. So I want to do a little history to say, how did we get to the point that we could decode a genome of an organism, 5 million letters, understand it, make diagnostics, and do this literally over a 24-hour period? Well, it starts with Frederick Sanger in 1976, who worked out the first way to decode DNA. And he did it in a clever way. First, he used new technologies that had given rise to the biopharmaceutical industry, the recombinant DNA technology, to get multiple copies of a DNA of interest, break it down, separate it by gel electrophoresis, and through autoradiography, determine the sequence. About a decade later, people wanted to use this technology to sequence not 5 million bases of an organism, as I described for that bacteria, but about 6 billion, the 6 billion that make up our genetic code as a species. And that project was really enabled by an advancement on Sanger technology, which had 2 parts. One was very Henry Ford-like, automating the sample preparation, automating how you take pieces of DNA, put it into bacteria, and grow it up. And the second was based on removing the radioactive labels and making them fluorescent, and then using automation to detect the ladder of DNA. And this technology scaled up at about 20 major sites around the globe over a few-year period, and spending about $3 billion, was able to give us the consensus human genome and really give us a map. At the time, I was actually working with Joel Bader, and our goal was to use this information as fast as we could from this consensus human genome to understand targets for drugs for cancer therapy. And while we were doing that based on the consensus, and for you, those of you who are entrepreneurs, I can tell you I felt I was on top of the world. I was CEO of a public company worth more than American Airlines when airlines had value. And we were mining the human genome. We had collaborations with large pharmaceutical companies around the world, Bayer, Roche, Glaxo, Genentech, Biogen. We were mining the human genome. We were finding drug targets. We were producing potential drugs. We were putting them into clinic. And I thought all was well. And I thought I understood the ramifications of sequencing a consensus human genome. Well, it turns out I was wrong in terms of my position, because on July 2nd, 1999, my son Noah was born, and he was rushed into the newborn intensive care unit, and he was having difficulty breathing. And at that moment, I recognized that I wasn't really interested in mining this consensus or canonical human genome. And I wasn't really interested in a map that described all of us. I really cared about Noah, and I wanted to know his genome, his personal genome, and I didn't want medicine that was good for cancer in general. I wanted a precision medicine that would help my son. And at the time, there was a magazine out, and that magazine had a picture of a computer chip. It was a Pentium 3 chip. And on that chip, they were bragging, they had 10 million transistors. And I had thought a lot about DNA sequencing up until that date. I'd gone to a lot of presentations that had talked about DNA sequencing. And all of those presentations had been based on taking what I showed you earlier, Sanger sequencing, and first automating it, as they did with fluorescence, and then trying to put it on a substrate. And I realized that, as a student of the history of science, that that was probably the wrong approach. What everybody wanted to do, and had literally spent tens or hundreds of millions of dollars trying to do, was take the gold standard, Sanger sequencing, and make it something that could be miniaturized. And I knew that the microelectronics industry did not come from taking the gold standard and miniaturizing it. The gold standard at the time was the vacuum tube, but the vacuum tube didn't scale well. The breakthroughs that Kirby and Noyce gave us came from taking an inferior technology, the transistor, because the transistor could be put on a single substrate, and you could both miniaturize and connect things with their monolithic idea. And so I thought about it. I recognized I probably wasn't better than the guys that had tried to move Sanger sequencing. So instead, I would take an easier approach. I would take an inferior technology, and at the time it was called pyrosequencing, and it was a little bit of a misnomer. Pyrosequencing actually didn't sequence. What it did was allow you to determine a single nucleotide by saying whether a nucleotide had been incorporated into a growing DNA strand by detecting through chemiluminescence the incorporation of a nucleotide. But what I liked about that is it was something as a chemical engineer that I could miniaturize. And when I miniaturized it, I would be able to, with a group of people, allow it not only to do a single base pair, but multiple base pairs. And we would be able to do it simultaneously. And that was one of the problems, if you will, with sequencing. You had to decode a lot of sequence. But the second problem, if we go back to that Human Genome Project, was most of the money of that Human Genome Project was actually spent breaking DNA into 300 or 500 base pair fragments, putting it into a vector, moving that vector into a bacteria, growing up that bacteria, and maintaining over 600 million colonies of bacteria. And so the second thing we wanted to do was get rid of that sample prep problem. And for that, I relied on a paper by Andrew Griffith, who had published in 1998 in Nature Biotechnology, an amazing, simple way to get clonality. What Andrew did is he just fragmented DNA and put it in an oil and water emulsion and emulsified it. The DNA went into the aqueous phase. And Andrew did something really fantastic. He actually put all the ingredients for coupled transcription translation, where DNA would go to RNA and RNA would go into protein, in that aqueous phase. And so he was able to do molecular evolution with millions of fragments of DNA. And I actually carried this paper around, I love it so much, for a few weeks. Again, before the iPhone, I actually carried a piece of paper around with the paper. So what we did is we said, look, this had been solved, the clonality problem, and all we had to do was add amplification to it. And that was pretty straightforward. We actually looked for surfactants that would survive thermal cycling, and instead of doing a coupled transcription translation, we would take a genome, literally break it up through sonification, put those pieces with some conserved ends into an emulsion, and PCR cycle it. Well, we went through all the available surfactants with no luck. But we did find a reference to surfactants that had been designed to be heat stable, and they were actually developed for explosive work during the Gulf War. And so instead of going to the cosmetics industry, we ordered these surfactants that had been developed for explosives, and immediately we got put on a watch list. But the good news is, it is one of those rare times when an experiment works first time. When we used the proper surfactant and we created these emulsions and we thermal cycled them, we were able to copy individually millions of fragments of DNA where each would be clonal. And we simply had a bead in them and we captured them onto those beads. At the same time, there was a group at Johns Hopkins that was actually doing this for genotyping and independently developed emulsions. The difference from us and the Johns Hopkins group was our emulsions were a lot larger, so we could support enough DNA to do DNA sequencing. And that was the Vogelstein group that had developed it for SNPs. So we sequenced our first genome. It was about 30,000 bases from a virus. We immediately put it into a gene bank, and we had two responses. One, this is going to be big, by Richard Gibbs, so we immediately put him on our scientific advisory board. Many of the students should appreciate that. And the second was from Edward Rubin. Since we had done a virus that was only 20 or 30,000 base pairs, he said something that established experts like to do, which is say something negative, because then you get a lot of press. And he said it would not scale. So as an entrepreneur, this is some advice, and you should follow it. We avoided Edward Rubin. So as an entrepreneur, it's great to have a technology, and that's an invention. But as the economists will tell you, you really want an innovation, which means taking your invention and having an economic impact. So we manufactured the machines, and we sold them, and we promoted them, and I'll talk to you about how we promoted it. We promoted it by trying to find the best scientists in the world that had the most exciting projects and showing our technology would work for them, and I'll go through some of those projects. But one of the nice things about a new technology when you get it out there, as opposed to keep it to yourself, is it has widespread impact. So people visited us that for the first time wanted to sequence literally gut. We didn't know what that meant, but now the microbiome is its own field. There was people from Cold Spring Harbor that said there were these teeny pieces of RNA that were interesting. At the time, there was no such field as microRNA. So an entire field blossomed from that. Metagenomics, ancient DNA, and other areas. There are literally tens of thousands of papers now leveraging this technology. I'm going to talk about being an evangelist. So it's great to develop a technology, but you got to get people to use it. And so we literally made cold calls, and that became my primary responsibility. And one of the groups we contacted had the first new drug for tuberculosis in 40 years. And that could be a big deal, because as you know, there was resistance to many of the current drugs. So we contacted J&J, and they sent us a number of bacteria, some that were resistant to the drug, and some that were sensitive to the drug. And to give you some historical context, at this time, if you sequenced a genome of a microorganism, you would literally have the cover of Science or Nature, and you probably had a budget somewhere between $10 and $40 million to do it. So what we proposed is that we would actually sequence each of these microorganisms and compare them to find out why some were resistant and some were sensitive. And our hypothesis was pretty simple, that the easiest way for a microorganism to become resistant to a drug is by mutating the target of the drug. That was the hypothesis. And that was important for the pharmaceutical company because when they want to develop a drug, they'd like to know the target so they can take a more rational approach to fast followers of the drug or perfecting the drug. So at the time, there was no software literally to do assembly publicly. And so we put together a group. Gene Myers, a leader in the field, came and advised us. Another great advisor, but we put him to work. He actually coded. And we actually wrote our own software to do assembly. We wrote our own software to do analysis. We did this project for J&J. We sequenced it. And I learned something. J&J learned the target of the drug and got a science publication. I learned that we weren't an author. And I was a little upset since we had invented the technology, we had done the project, we had written the software, we had found the mutation. I was trying to figure out what we hadn't done. But when I complained, they said, no, if you look at reference 15 or 16, we do acknowledge that work. So that was our first experience with co-publishing with a large pharmaceutical company. At the time, there was a beautiful science publication by Matthew Meyerson, who's at the other medical institution in Boston. And Matthew had a beautiful paper, and it basically said that patients taking Iressa, who had a mutation in the target for Iressa, wouldn't respond, and kind of suggested in his paper that when you do a clinical trial, you should probably exclude those patients. So I called Matthew, and I said, that's going to change the entire pharmaceutical industry. And he said, no, Jonathan, they're ignoring it because of the pharmaceutical industry. For the most part, you get your bonus by passing something along, not by killing things. And this is actually true, literally motivated to push things further along, whether they're good or bad. And it actually took probably close to a decade for the pharmaceutical companies to realize that you should do genetics during your drug development. But Matthew said he would like to use our technology immediately to help his patients. And his problem was, when you Sanger sequence and you take a biopsy, you only see mutations if they're prevalent at greater than 30% of the tumor. And unfortunately, by the time a mutation that's resistant to a drug is 30% of a tumor, it's probably too late. And so what he wanted to do is see if he could see mutations early on in these biopsies and switch the therapies to something that would be more effective. So instead of sequencing a whole genome, as I've been discussing, we decided to sequence the same section of a genome over and over and over again, literally hundreds of thousands of times, to detect a mutation early on. The patient Matthew had was actually a scientist who could give an informed consent because they understood what we were doing. And Matthew was able to show a mutation and was able to switch the therapy. And so I think this is the first example of precision medicine by deep sequencing. And the patients then had a positive response. I was pretty excited, ran back to 454, said, you know, we're saving lives. My lawyers immediately required us to put with every machine we sold and every kit we sold the letters research use only all over it and not promote it for diagnostics. The good news is physicians, for literally a decade before sequencing was approved for uses like cancer diagnostics, were able to do it under their own authority before the approval. But that was the first example of deep sequencing and changing a therapeutic. And now all major cancer centers will sequence a dozen or a few hundred genes and determine what therapy you should get. If they don't see mutations in that dozen or few hundred genes, at a number of centers, they'll actually sequence all 25,000 genes and do full exome sequencing. And it's now part of the standard of care for cancer. Another quick study that we did was for HIV resistance, very similar to the biopsy case. You want to change the medicine when somebody's resistant to HIV before the resistant virus is the dominant virus, because by that time you've destroyed the patient's immune system and they're not going to have a good quality of life. And so we did some early work on HIV resistance, and the industry as a whole, as you know, has done just a fantastic job and has really, through understanding resistance and combination therapy, have really changed AIDS from a death sentence to a chronic disease. This is a work that's probably most important because it's the only work I ever did that got us on a movie set. And this was work with Ian Lipkin. And Ian was actually an advisor to Contagion and actually put the 454 machine on the Contagion set. But Ian had a real problem. And the problem was that patients who had received organs, all from the same donor, all died. And Ian sent us samples from each of these patients. And we just started to sequence and create a database. And when we did that, we found a virus that was normally not lethal, was lethal when people were under immune suppressants that they have with organ transplants. Didn't help these patients, but it does help future and has helped future patients because now you can check for this virus before you receive a transplant. But this is a pretty important lesson for me on both the genetics, what is the virus, as well as the environment. And that's a theme that anybody doing complex diseases will recognize. But I saw it in many circumstances. So here is another study we did with Ian Lipkin, where his collaborators had literally collected the jelly from colonies of honeybees that had collapsed and from colonies that hadn't collapsed. And again, he sent us the nucleic acids, and we literally started to just create a database of all the nucleic acids that were in colonies that had collapsed and ones that hadn't collapsed. And lo and behold, we found a virus in the colonies that had collapsed. But before people run off and say, oh, now you know what's killing the honeybees, I think this is very, very analogous to the situation with the transplant patient. In that this virus could have been just fine if it wasn't for other environmental stresses that have put the honeybee in a position that this virus collapses it. So it's very, very important when you do these things. You can find something, and you can even fulfill Cox's hypothesis. They actually take this virus, put it in a honeybee colony, and it collapses. That does not mean it's the cause of the collapse. There are environmental stressors that somehow are making the honeybee susceptible to this virus, and people are working on that now. So I think I'll tell a second personal story. I said I got into DNA sequencing because of my son Noah. Well, it turns out I was interested in sequence long before I had my son, and this is hard to corroborate, but I was interested in sequencing dinosaurs before the movie Jurassic Park. I kid you not. And so when we had a way to sequence, and our sequence was based on grabbing a single molecule, putting it in this emulsion, and amplifying it, I thought it might be perfect to sequence where you don't have very many molecules of DNA, and when they're already fragmented for you, ancient DNA. And I had been familiar with some papers that had been published, I think both in Nature and Science, showing that people had insects from amber and were able to sequence DNA that was 23 million years old. So I'm figuring, look, if they could sequence a little DNA by Sanger and get 23 million years, I'll sequence a lot of DNA and get 65 million years, and I'll have my dinosaur. So I called Svante Paavo, and he was the world's authority on ancient DNA in Neanderthal, and said, let's sequence some dinosaurs. He didn't hang up on me, but he explained to me, and this was just a total shock, that everything published in Science and Nature is not necessarily true, and that all of those reports were false, and you can't repeat it. And no, you cannot sequence DNA that's 23 million years old from amber. I didn't even know what to say at this point. So I said, well, send me Neanderthal DNA. And then he explained to me, well, that's probably the rarest DNA on the planet. I have graduate students that spend their entire career trying to get femtograms of this DNA, so I don't think I can do that. However, I have pounds of cave bear DNA, and it's 35,000 years old, and the last Neanderthal samples we have are about 35,000 years old, and I can send you that. And that should have DNA fragments from 60 to 120 bases, and let's see what you can do with that. So he sent us the cave bear DNA, and again, one of these experiments that works for first time, we sequenced that 454, the DNA from the cave bear, 35,000 years old, and at that moment, we had more sequence of ancient DNA than had been sequenced cumulatively before. It got Svante very excited, and he got on a plane, and he did two important things for research. He came and gave us the Neanderthal DNA, but he did the second most important thing for research, which is he got the funding, and it was the 150th anniversary of the find of the actual Neanderthal in the Neanderthal Valley. And so he got the Max Planck to write us a check for $5 million to sequence it. And that's what we did. And again, I thought I was on top of the world, sequencing Neanderthal. I rip open my New York Times to read about it, and all it says is George Church says we can now clone Neanderthal. Anyhow, it was still good work. And George is a friend, and at the time that I was telling my son about this work, another group published Wooly-Mammoth. And so at first I said, we're scooped, but then I noticed in all the PR photos are machines, so I felt okay. But then when I went to read about it, all of them said, George Church says you can now clone Wooly-Mammoth. So, and you probably can. There's only 400 amino acid differences between the Wooly-Mammoth and modern elephant. I don't know if it's the amino acid changes that make these beasts, but you could start doing some mutagenesis, maybe with CRISPR, and actually come up with a woolly mammoth. And that's probably better than the Neanderthal project. At the time, a lot of people were talking about human variation, and you probably remember after the consensus human genome, tremendous amount of popular press saying we're all the same. And we did an early experiment with Mike Snyder and others showing that it's not just about base changes. In fact, by using big fragments of DNA, looping them together, an idea I actually borrowed from Bender from Harvard, who had done it for flies, we did it in man, and we were able to show a huge number of differences between each of us by giant sections of DNA that are moving around. So at this point, I figured it's time to finish the project. We started the company because we wanted to do personal genomes. We thought the best genome to do was Watson. I couldn't exhume Crick, so met with Watson, got his blood, and sequenced him. We published it. Took over a year to get it published. I had a lot of ethical lessons at that point. We were actually rejected on ethical grounds. I truthfully didn't even know what that meant, but my paper was rejected. We literally did something right out of a Woody Allen movie. How many of you have seen Annie Hall? Perfect. Do you remember that scene where there's somebody babbling about some book, and Woody Allen grabs, I think, Marshall McLuhan and pulls him on the stage, and he tells the Columbia professor he's an idiot, and then goes away? And he says, "Don't you wish you can do that?" Well, that's precisely what we did. We got the foremost medical ethicist from Baylor. She wrote this beautiful rebuttal that they published in Nature along with our paper. To this day, I don't understand her rebuttal, but we got it published. So the good news is we had made a new way to sequence. Since you're entrepreneurs, our first full year of sales, we sold over $100 million worth of stuff. Again, you think you're on top of the world. But I found out I was actually out of a company. A large pharmaceutical company had come in, bought my company, and I was fired. So that's pretty tough. And I had kids, so you can't let them know it's tough. So you just got to dust yourself off, stand up, and rethink things. And one of the reasons I felt I was on top of the world was I couldn't go anywhere where I couldn't make a friend, because everybody wanted to sequence something. And that's really great. You get to call scientists, you get to do the most fun projects in the world. And so I knew I needed a sequencing technology. And I really wanted to democratize sequencing. That was just a start. I wasn't finished. But I would no longer be first. And when you're first, it is easier. You're not competing with anybody. 454 had a monopoly for 2 years on next-gen high-throughput sequencing before Illumina and others caught up. So I had to rethink my strategy. I wanted to sequence, but I wouldn't be first. Also, I wouldn't have the resources of other companies. Companies at the time had literally raised over $500 million to enter the sequencing market. And so I had to rethink, and what I decided to do was leverage the electronics industry. So I told you I got my inspiration for 454 and our next-gen sequencing from that monolithic idea, which gave rise to semiconductors. But now I wanted to not get inspiration. I wanted to get literally their supply chain, their factories. Over $3 trillion went into that supply chain. And I knew to compete with those people that had $500 million, that had market share, I would need an advantage. So my advantage was microelectronics and that supply chain. And I knew that anything they touched, they dominated. So the first cameras I know people laughed at, and they said they're not as good as film. but I literally watched on my iPhone the announcement of Kodak going bankrupt. I just realized there's people in this audience that don't know who Kodak is. They used to make film. So anyhow, semiconductors win. And we all saw semiconductors win, first with our personal computers, and now with our portable electronics. So my goal was pretty simple. I wanted to sequence using semiconductors. And the idea was you have biological information. You have chips right now that see photons, right? That's how you take a picture with them. And I simply needed to make a chip that would see chemistry or ions. And so all of you are familiar with this. This is just a strand of DNA and making a complementary strand of DNA. And this is the basis of next-gen sequencing, where you detect this base by either having a different color label on it, by detecting the pyrophosphate and doing chemiluminescence. But what I figured is we would just detect the hydrogen ion. And all of us have detected hydrogen ions. Raise your hand if you used a pH meter in the last 15 years. Okay? So pH meters, when I first used them, had a glass electrode. But over the last 10 or 15 years, they've switched to have an electrode that's based on a transistor where the gate is floating and becomes the reference electrode, and you detect that hydrogen ion through this transistor. And so I actually had thought about this in 1993 when I was starting my first company. But at the time, there was no easy way to get millions of these on a single chip. But when I went back and looked at it in 2006, what I found was a publication from 1999 where a Spanish group had taken these things known as ISFETs and had made them on CMOS. Those 4 letters are magic. So CMOS is the specific semiconductor technology or foundry process that gives you all the parts of this electronics. And once something can be done on CMOS, I knew I could do it at incredibly low cost and in a fantastically, massively parallel way. And so I put together a group of people, and we simply took ISFETs and made a chip that had millions of them that we could read out, and put little wells on top of it and sequence on top of it. And we were able to build a sequencing machine where you had a chip, and we could sell these chips for $99. So this is the chip that was used to decode that bacteria I talked to you about in that outbreak in Germany. And in fact, you wouldn't decode 5 million base pairs. You would detect or decode 500 million base pairs, stitch it together, have 100-fold redundancy, so you'd have great accuracy. And it worked by having that chip with little wells. And when I talked about 454 sequencing, I used to hold up a glass slide and say we could sequence 4 pieces of DNA on the end of a human hair. And now we would be sequencing 400 bases or pieces of DNA on the end of a human hair. And we could do it in a massively parallel way. And you can do it in 2 hours instead of weeks, like some of the techniques that use fluorescence take. The nice thing about semiconductors is you can go to newer and newer factories and get higher and higher densities, both of the electronics underneath to read out the sequence, as well as the containers, if you will, for the sequencing reactions. And now we have chips that for $1,000 can do a human genome, and they do about 14,000 DNA sequences on the end of a human hair. I remembered my first criticism when we did the first next-gen sequencing of a virus. It won't scale. So this time when we published a human genome, and just in case anybody came out of the woodwork and said it wouldn't scale, we sequenced Gordon Moore's genome, who gave us Moore's law, the ultimate authority in scaling. It was also the first non-light genome, because prior to that, you had always had either radiation and or light. So it does scale. And it was a great opportunity for my son to meet Gordon Moore. He's an amazing guy. Before I start wrapping up, I want to talk about a misconception that I hear a lot. Over and over again, I go to presentations and I see slides that say DNA sequencing is moving faster than Moore's law. I'm here to tell you it's an illusion. We're just catching up to Moore's law. And once we catch up, we're going to face the same bottlenecks everybody else does, and we'll advance with Moore's law. What do I mean by that? Well, if you look at 454, it was at a density that was equivalent to semiconductors in the 80s. If you look at the first chips we did at Ion that did that bacteria, 5 million bases, did it in 2 hours for $99. Well, if you look at the new chips, they're simply made at a foundry that's about 10 years more advanced. If you look at anything that came out of those two foundries, you'll see a 1,000-fold increase in performance. And the nice thing about a 1,000-fold increase in performance is that's the same difference in the size of a bacterial genome to a human genome. And so by moving from 1990s foundries to make these large wafers that are then cut up and make the chips, you just make them, in a newer foundry, and you get the $1,000 genome. I'm going to do some acknowledgments, and then I am going to do a little what's next promotional section. But I want to acknowledge the genomes that we got and the great people that I get to work with, Watson, Gordon Moore, and Charles Towns. I'm pointing at this with a laser. Charles Towns recently passed away, and he is the father of the laser. And this is a collaboration I did with my niece at Burning Man, where we sequenced Charles Towns' genome, and she beamed it into outer space towards hopefully a habitable zone planet 25,000 light years away. And so I'm convinced that if we're ever invaded, this is what they'll look like. What's next? When we did sequencing, we had this vision of it being all over the world, information being aggregated, great computer systems being able to analyze it. But even as recently as when we founded Ion Torrent, we were unable to do it. At the time, there was no such thing as a HIPAA-compliant cloud, a cloud that would meet the regulatory requirements of the healthcare system. But now there is. So what we're doing now is we have an incubator. We call it 4Catalyzers. And we're creating a number of companies, all with an identical strategy. They're all creating devices that will advance the technology 100 or 1,000-fold in terms of cost and ease of use. All of them are designed to put the information into the cloud. It was wonderful seeing the presentations today, and you could see how easily people can do that now. And in our case, we have a group that's using a new technology. For those of us who have followed AI for 30 years and been disappointed, it's all changing. Literally, in the last 12 months, these deep learning algorithms have done what couldn't be done before. So if you look at image recognition, they're now as good as people. And if you think about big problems, like taking pictures of galaxies, where they actually have hundreds of millions of pictures, humans can't sort through them, and these can. So this is probably the first technology that can keep up with the data that we can produce from these devices. And now you have this beautiful virtuous cycle where you have a device, the cloud, the deep learning, and your devices get smarter and easier and better. And I'll just give you a little preview of what we're up to. We have 4 companies. They're all building devices. We're leveraging smart advisors. We're going outside of our field to find people who have some of the best algorithms, if you will, who have some of the best computers. Both David Ferrucci and Max Tegmark are both advisors to our SABs. And I show this work that Joel and I did many years ago to say that machines don't just have to be silicon-based. So Lam Therapeutics isn't using a machine or a device in the sense that most of you are used to. It's using transposons or elements that can go around a genome. And We still use the same strategy, though. You accumulate the data, and you analyze the data. We're looking for people that love what they do. One of the most exciting things that I found recently are there are fantastic people that aren't as smart as you guys. You guys knew that the best thing to do right away was biomedical research because you can have impact on people you love. A lot of people haven't been well advised. They're making weapons. They're trying to beat the stock market. And we've made a big effort to recruit these people and get them to work with people like yourselves because they have fantastic skills. They're just doing the wrong thing. So that's where we are now. If you'd like to change the life of somebody you love, please send me your resume. We have 4 startups. We're looking for exceptional people. There isn't a skill we're not looking for. So whether it's single molecule biophysics, IC design, if you're passionate and you love what you do, and again, you really do want to have no regrets, work on something that could save the life of somebody you love. Thanks.